This is the fourth video in the fundamentals of biology, and today we're going to be covering enzymes and cellular energy. So first we have to talk about the two laws of thermodynamics. The first law is also known as the conservation of energy, and it basically states that energy is not created or destroyed, and the energy is only transferred. And energy is basically the capacity to cause change, such as kinetic energy, potential energy, thermal, and chemical energies. So you can see in this diagram, chemical energy is being transformed to mechanical energy. However, there is no energy that is lost. Rather, it is transferred. And same thing with this apple falling down a tree. Potential energy is being transformed into kinetic energy. In the second law of thermodynamics, it says that every energy increases the entropy of the universe. And what entropy is basically the molecular disorder or randomness in the universe. So entropy within a system can still decrease as long as the entropy of the system and the surroundings, which basically makes up the entropy of the entire universe, increases. So you can see in this block of ice, as it freezes, the entropy will increase, which is spontaneous and will occur without the input of energy. However, if you're trying to freeze it back, it will not be spontaneous and will require energy, but the net entropy in the entire universe will still increase. So when we're talking about spontaneity, we're really talking about Gibbs free energy, or the total energy able to do work. So the equation of Gibbs free energy, or the change in G, is equal to the change in enthalpy minus the temperature times the change in entropy. So enthalpy is the change in the total energy, whereas Gibbs free energy is the energy that is actually able to do work. And you have to subtract the temperature in Kelvin multiplied by the entropy, which we just talked about. If the change in S is positive, you have increased the entropy. So as a result, the change in G, or the Gibbs free energy, will be negative. And as a result, that will be a spontaneous reaction. On the other hand, if your change in S is negative, you have decreased entropy. So the change in G will be positive since you're subtracting a negative number. And as a result, this will be non-spontaneous and will not happen normally without the input of energy. And if your initial, and during conditions where your initial Gibbs free energy is positive, it's going to be very unstable and wants to undergo a spontaneous reaction. So if you just look at this clumped up molecules, they're going to be pretty unstable, and as a result, they want to do work in order to become more stable. And what they will do is disperse. And as a result, the G will decrease, and as a result, the Gibbs free energy change will be negative. And yeah, these molecules are all going to be more stable than they were before. So now let's talk about ATP. ATP stands for adenosine triphosphate. So you can see that it is made up of a ribose sugar, a adenine nitrogenous base, and 3 phosphate groups. So how does ATP work? When it is hydrolyzed, a phosphate group will attach to a recipient, and that will leave ADP as a result. So you can see here, when ATP is hydrolyzed and you take out the water, it is going to result in ADP, and it will leave a phosphate group. And this phosphate group is going to attach to the molecule that it wants to speed up the reaction for. And this will make it, the unstable molecule, a phosphorylated intermediate, which is unstable and capable of work. So you can see a previously non-spontaneous reaction will be able to occur. And this process of using an exergonic reaction to drive an endergonic one is called energy coupling. And so after the phosphate group is able to reattach to the ADP, it regenerates ATP. And that's why you're able to regenerate and have a renewable cycle of energy for ATP. So now we're going to be talking about metabolic pathways, or the chemical reactions to get from one molecule to another. So from the reactants to the products. And we'll be comparing endergonic and exergonic reactions. So in an endergonic reaction, the entropy change will be negative, whereas in the exergonic, the entropy change will be positive. If we're comparing the Gibbs free energy, the change will be positive in an endergonic, which means that's non-spontaneous as we have shown before, and in an exergonic, it will be spontaneous. And this is because in an endergonic, you will have to input energy or use ATP, whereas in an exergonic, you will be inputting energy, and it creates ATP from an ADP and a phosphate group, or an adenosine diphosphate with 2 phosphate groups. And in an energonic reaction, it is anabolic, which means it is building into a larger substance. In an exergonic reaction, it is catabolic, which means it breaks down to smaller parts. And that means in an energonic, the product will increase in complexity because the bonds are forming, whereas in exergonic, the product decreases in complexity as you break them into smaller parts. So you can see the Gibbs free energy charts for both the energonic and exergonic reactions. And you can see how the energonic will increase, making the change in Gibbs free energy positive, whereas in an exergonic reaction, the products will have a lower Gibbs free energy than the reactants, and energy is released. So as a result, the Gibbs free energy will be negative. And now let's talk about enzymes and catalysts and how they work. So just some basic terminology: a catalyst is anything that speeds up reaction, and an enzyme is a specific catalyst, that is a protein. And catalysis is the process of speeding up a reaction. So just basically how an enzyme works. So how do enzymes work? They basically lower the initial energy required to start a reaction. And this energy to start the reaction is called the activation energy. So you can see in a normal reaction, you're going to need a lot of energy to overcome in order to produce a final product. Whereas in a reaction that has an enzyme included, the activation will be lower. So you're going to need less energy to break the initial bond to release into the final product. And activation energy is usually things like thermal energy. So what happens when enzymes work? So an enzyme will act on a substrate, which is the reactant, and this result will be the enzyme substrate complex. So this substrate will fit onto the active site of the enzyme through induced fit. And that basically means that it is a specific shape, and that substrate has to fit onto this enzyme. And after, the products will release. And the result of enzymes is that they will orient the substrates and create a favorable environment. And this is why the reaction will go faster. And the rate will actually increase until the entire reaction is saturated, which means all the active sites are used up. So you can see in this diagram, if you're increasing the number of reactants, the rate will keep increasing because you have enough enzymes. However, once there are not enough enzymes to accommodate all the reactants, the speed will start to stagnate. And now let's talk about specificity and denaturation of the enzymes. So, as I have said earlier, enzymes are very specific, and we call it a long key fit because the active site has a very specific shape that will only allow specific substrates. And this is most likely why there are very specific enzyme names for certain substances. So sucrose, you're going to break it down through something called sucrase. So the ACE basically shows that it is an enzyme. And things that can cause denaturation or a change in the shape is the temperature and the pH. And what they do is they change the shape of the enzyme and the active site. So you can see in this diagram the optimal pH for two different enzymes. So for pepsin, it will work the best at a pH of 2. If you try to increase or decrease the pH, it will not work as well because the active site will be deconformed. So basically, the takeaway from this is each enzyme has an optimal temperature and pH to most effectively lower the activation energy, and we call those optimal conditions. Now let's talk about things that can affect how an enzyme functions, and these include cofactors and inhibitors. So cofactor is any non-protein enzyme helper, and an example of this would be metal ions. However, if this cofactor was organic or made of carbon in biology, it would be called a coenzyme. An example of this would be vitamins. And inhibitors are things that basically inhibit how an enzyme is able to function. In competitive inhibition, they will bind to an active site to compete with the substrate. So you can see in this diagram, a competitive inhibitor will block the active site. So the original substrate that wanted to get on the enzyme will not be able to bond. However, in non-competitive inhibition, it will bind elsewhere to change the shape of the active site. So a non-competitive inhibitor will not bind to the active site, it will instead bind somewhere else on the enzyme, which changes the shape of the active site. And this all comes down to allosteric regulation, which is the binding of one site to affect the protein or enzyme function at another site. So allosteric activators stabilize the active form of a protein or enzyme, and this turns the protein on, and cooperativity amplifies the enzymes. Whereas allosteric inhibition stabilizes the inactive form and turns the protein off. And the reason we need these on and off switches is because we don't want a protein always happening, or else it's going to take up too much energy. And what feedback inhibition does is it shuts down a metabolic pathway by producing an inhibitor as part of the end product. So you can see in this diagram, the final product will actually have something that inhibits the active site. And as a result, the process of creating whatever the enzyme or protein wanted to do, it will end. So thanks for watching. Today's video covered thermodynamics and energy, spontaneity of reactions, enzyme function, and enzymes regulation.